Hello everyone, Mr. Linder here. Let's talk about the complement system. The complement system is a collection of more than 25 plasma proteins and cell membrane proteins that, when activated, aid in immune processes such as opsonization, inflammation, and production of the membrane attack complex. The complement proteins, many of which come from the liver, are secreted in inactive forms and must be activated through several pathways. The pathways include the classical pathway, the lectin pathway, and the alternative pathway. And these pathways are used to generate a protease called C3 convertase that ultimately creates complement fragments that are used in nonspecific immunity. Since the complement system can be extremely damaging to host cells, there are complement control proteins on the surface of cells and in our blood plasma that prevent the activation of the complement system. The most important inactivators are C1 inhibitor and the CD59 protein found in the cell membranes of host cells. The lectin pathway is actually very similar to the classical pathway. So I'm going to discuss the classical pathway and just understand that the lectin pathway is very similar. Then we'll take a look at the alternative pathway as an alternative mechanism for creating C3 convertase. So let's start with the classical pathway. The first protein in the classical pathway is the C1 complex. The C1 complex is really a group of 3 proteins in which the final protein acts as a protease. What's interesting is the C1 complex can actually bind directly to pathogen surfaces, and it can interact with antibodies. So the C1 complex can actually bind to IgG antibodies and IgM antibodies. IgG and IgM. So that's one way to start the classical pathway, is to have the C1 complex interacting with antibodies that have already bound to the pathogen surface and began to tag it for destruction through now non-specific complement pathways. The protease of the C1 complex can be used to activate other proteins. So the C1 complex protease can actually activate the C4 complement protein and the C2 complement protein. Typically, when complement proteins get activated, they're broken down into 2 fragments. So C2 becomes the C2A and the C2B fragments. C4 becomes the C4A and the C4B fragments. The designation B represents the larger fragment. Therefore, A would be the smaller fragment. And it's typical that the larger fragment is going to interact with other proteins and go on to activate something else within the complement system. So for example, when C4b and C2b join together, they form the C3 convertase. So here we have C4b and C2b, the larger fragments when C4 and C2 were activated, and this is known as C3 convertase. This protease, this enzyme, is going to be used to activate the C3 protein. So the idea behind the classical pathway is to create C3 convertase so that the protein C3 can be activated. When C3, this protein right here, when C3 becomes activated, it separates into two proteins. So we get the C3A fragment and the C3B fragment. The purpose of these fragments is to initiate non-specific defense mechanisms. C3b is a very good opsonizing protein, so it's involved in opsonization. Opsonization is where we can tag pathogens to make it easier for phagocytosis to take place. The C3a fragment is going to be involved in the inflammatory reaction. So C3a causes inflammation. What's interesting, though, is there are other complement proteins that are also involved in the inflammatory reaction. And the inflammatory response is to trigger the release of histamine, to increase vascular permeability, and to enhance chemotaxis, which is to draw white blood cells to an area of the body where a pathogen has been invaded. So what else is going to be assisting in inflammation? Well, what's interesting is that C4b and C2b can also combine with C3b. So we actually have C4b and C2b, those two larger complement fragments that were used in the C3 convertase. It turns out that they actually interact with C3b to form another enzyme. It's called C5 convertase. This particular protease enzyme is used to activate the C5 protein. So C5 convertase is going to activate the C5 protein. When C5 becomes activated, it splits into C5b, the larger fragment, and C5a, the smaller fragment. So it turns out that C5a is going to be working with C3a in the inflammatory reaction. And so again, those complement proteins are being used to trigger the release of histamine and to lead to vascular permeability and chemotaxis in what we see as the non-specific inflammatory reaction. The C5b protein, which is the larger protein, actually combines with 4 other complement proteins. So C5b is going to combine with C6, C7, C8, and C9. So these other 4 complement proteins to form what's called the membrane attack complex. So the MAC, or membrane attack complex, is actually made up of these 5 complement proteins, starting with C5b, C6, C7, C8, and C9. And what these complement proteins do is they insert themselves into the wall of the pathogen. So if this is the target pathogen, what these complement proteins are doing is they're inserting themselves into the wall of the target pathogen, creating a hole in the pathogen so that the target pathogen will lyse. So these pores throw off the osmotic balance, and they're going to cause the pathogen to lyse. The alternative pathway is exactly how it sounds. It's an alternative way to get to C3 convertase. So the alternative pathway is really just another way to activate the C3 protein. What's interesting about the alternative pathway is that this process can occur on the surface of pathogens without the presence of specific antibodies. So we saw in the classical pathway that the C1 complex can actually interact with IgGs and IgM in order to start that classical pathway. The alternative pathway doesn't need specific antibodies to start the pathway moving forward. What's also interesting is that the C3 protein can actually spontaneously separate into C3a and C3b. So there's actually a scenario where the C3 protein is actually converting into C3b and the C3a, and it turns out that you need C3b to create an alternative form of C3 convertase. So once you get spontaneous conversion of C3 into C3b and C3a, the C3b can attach to the pathogen membrane. So let's say, for example, this is the pathogen. You'll have C3b attaching to the pathogen membrane, and that's going to start this alternative pathway. C3b binds with factor B. This is a plasma protein that is part of this alternative pathway, and it's going to interact with an enzyme called factor D. Factor D is the necessary enzyme for splitting the B protein. So factor D is an enzyme, it's a protease, that's going to activate the B protein, and so B is going to split into B sub A and B sub B, with the lowercase b, again, meaning that it's the larger fragment. The combination of C3b and the larger B fragment is what makes the alternative version of C3 convertase. Because remember, the classical pathway used the C4b protein and the C2b protein in order to create C3 convertase. So this is an alternative version of the convertase enzyme. Once you have C3 convertase, this will take the C3 complement protein, split it into C3b and C3a, and we are off and running in essentially the remainder of the classical pathway in order to create the non-specific defense mechanisms of opsonization, the membrane attack complex, and the inflammatory reaction. So the classical pathway gets the activation of C3 convertase using the C1 complex, and the alternative pathway activates the C3 protein using a C3b that is spontaneously created and the activation of factor B. If you also notice the factor P protein, the factor P protein is really a stabilizing protein for C3 convertase. So it allows that enzyme to remain stable so that it can activate the C3 complement protein and allow us to get to opsonization, inflammation, and the membrane attack complex. So that's a little bit on the classical pathway and the alternative pathway for complement protein activation. I hope that helps. Take care.